Hello everyone, I'm Mike Kettles, the Executive Director with Cure Glaucoma Foundation, and we're joined today by Dr. Thomas Johnson III. Dr. Johnson is a glaucoma specialist with a clinical practice at the Wilmer Eye Institute at Johns Hopkins School of Medicine. He's also a research scientist and has a translational science lab in the Smith Building at Johns Hopkins. The purpose of our discussion with Dr. Johnson is to find out how a grant from the American Glaucoma Societies MAPS award program helped his early research efforts. MAPS stands for Mentoring for the Advancement of Physician Scientists, and it's designed to help early researchers get funding in their initial efforts. So thank you, Dr. Johnson. We really appreciate you spending time with us today.
Of course, great to be here. Thank you.
Certainly. So I read a little bit about you, and I understand your dad was a veterinarian, and you kind of grew up around medicine and used to help him on Saturdays.
Yeah, that's right. He had a small private practice outside Chicago and would treat dogs and cats. And I loved animals, so I'd go and spend half a day or a day with him every now and then and play with the animals, but also kind of see how physical exams are done, laboratory work, looking at slides under the microscope, a lot of science and a lot of medicine, and appreciating the power of healing a little bit, where you could see animals that had been sick or cured, and I really wanted to be able to do that for humans.
I see. And so, but at some point, you decided you wanted to treat people, and then you got an undergraduate degree from Northwestern in biology in 2005.
Yep, that's right.
And then decided you were interested in trying research, so you took some time off to do that before medical school?
Yeah, exactly. So I did my undergrad, I had a lot of AP credits, so I happened to finish in 3 years, but I didn't then have my act together to apply to medical school just yet. So I took a year and did research at the University of Nebraska, actually. And I was working primarily with Carl Camerons, who was a glaucoma specialist that incidentally was credited with having discovered prostaglandin analogs as a treatment for glaucoma. Your patients will know this as latanoprost and some of the other medicines in that class. He was responsible for just making the discovery that those medicines lowered eye pressure. So I spent a year with him, and we were doing work on aqueous humor dynamics, trying to understand how aqueous humor, the fluid inside the eye, is formed, and why in some people it's not drained effectively from the eye, and why that leads to high eye pressure. And so those were clinical studies, but then I also spent a little bit of time doing some basic science research, looking at retinal tissue and doing some work with animal models of glaucoma to look for neuroprotective therapies, or ways of preventing optic nerve damage in glaucoma through novel pathways that weren't necessarily related to the eye pressure.
I see.
So I became so interested in that, and I felt that basic science research had such a potential for developing new therapies for eye disease that I actually deferred medical school, even though I applied to and was accepted at that point. Johns Hopkins was kind enough to give me a 4-year deferral so that I could go do a PhD in neuroscience.
I see. And so once you were done with that, then you went to medical school, and I guess you did your ophthalmology residency after that.
Yeah, I've been at Hopkins since 2010. I did medical school, internship, residency, glaucoma fellowship, and chief residency here at Hopkins.
Okay. And so looking at the MAPS award, can you talk a little bit about getting funding for your early research efforts and sort of what that landscape looks like? I understand it's kind of a struggle for researchers when they first start out.
Yeah, that's exactly right. I mean, when you are established and have an up and running lab, people count on NIH for funding and large project grants like R01s that give hundreds of thousands of dollars to support yourself and the people in your lab. But you can't just have a good idea to get an R01. It takes more than that. It takes preliminary data that will convince reviewers of your grant that what you're going to do is feasible and likely to work and produce results that could help people. And so you need to be able to do experiments and collect data for a significant period of time even before you're really eligible to apply for those large grants. And that's where funding from foundations and from the American Glaucoma Societies MAPS Award, supported by your foundation, has come in tremendously handy. I think for early stage investigators, there's a lot of demands on your time, and it's difficult to get data sometimes because on the one hand you need to be in the lab with your hands doing experiments, working with pipettes or microscopes or whatever it may be, but then you've also got a lot of administrative things that you need to take care of. And you really need to be able to hire people, technicians, students, postdocs to work with you to collect this data. All of that costs money. And the MAPS Award in particular is geared towards physician scientists who have an extra, I don't want to say burden, but challenge to face. And that is the fact that we're also physicians. So I treat patients with glaucoma. I have a full day clinic every Friday, and I'm in the operating room every Thursday. So the amount of time that I have to do science myself and to help my postdocs and technicians that are collecting data in the lab is even more limited. And if I didn't have MAPS funding, I would be required, or out of necessity, I'd be seeing more patients and doing more medical practice, which is great. But the MAPS Award and early clinician scientist funding protects the time of the physician scientist and allows them to take a step back from clinical work and focus on the science, to really think through their ideas, to collect the data, and then eventually get that independent funding that will support the lab for years at a time.
I see. When you applied for the MAPS grant, is that like a normal grant application where you sort of present a research idea and then simply compete with other people who are doing the same thing?
To some extent, you definitely need to come with an idea, and I presented, I think a 3 or 4 page plan of exactly what I wanted to do. It's reviewed by a team of scientists from the American Glaucoma Society that judge it based on its significance, its impact, its innovativeness, and how likely it is to help patients now or in the future. Unlike an R01, I didn't have to give a whole lot of preliminary data because that would sort of defeat the purpose. This is an award that would allow me to collect that preliminary data to get further funding down the road. But you definitely need to have a well thought out plan and scientific proposal with specific experiments, and then that's judged by the panel against all the other people that submit their ideas to see who will receive the funding.
I see. So then it basically sort of acts as a springboard. Once you get the funding from AGS, from the MAPS program, you're able to engage in the research and then use that to attract additional funding in the future.
That's exactly right. Yeah.
Okay. I'm also curious, so you have 2 roles. As you said, you're a clinician who sees patients, glaucoma patients. So that's the treatment end of all this. And then you're in the lab. Yeah. How does having a foot in both those worlds kind of inform each other? I
think it's tremendously important. The reason I'm doing the work that I am in the lab is totally informed by my experiences in the clinic. And I think it's most glaucoma patients have been told, and certainly all glaucoma doctors know, the key to glaucoma at this point in time is early detection and lowering the eye pressure so that the glaucoma doesn't get any worse. Glaucoma vision loss occurs when the optic nerve becomes damaged. The optic nerve is composed of a type of neuron called a retinal ganglion cell. In fact, most people are born with about 1 million of them per eye. And glaucoma causes the death of those retinal ganglion cells. And when those die, they're lost permanently. We don't regenerate those. So glaucoma doctors will often have to tell people there's really nothing that we can do to bring back the vision that you have already lost. And hopefully most patients are being diagnosed before they notice a tremendous amount of vision loss or before it has a huge impact on their life. But some patients come to my clinic being diagnosed for the first time with total blindness in one eye and only the ability to detect motions out of the other eye. And when you find the disease that late, there's unfortunately not a lot that modern medicine has to offer. So all of the work I'm doing in the lab is based on trying to develop the first treatment that will be capable of actually restoring vision in glaucoma or reversing blindness in this disease and really a lot of other optic nerve diseases as well. And that's through transplantation of stem cell-derived retinal ganglion cells to try to regenerate the optic nerve. So in one sense, what I do in the lab, or what I've experienced in the clinic informs what I'm doing in the lab, and I have a front row seat to understand the clinical potential of this therapy. And then eventually, when we're out of the lab and thinking about taking this in clinical trials to human patients, my experience in the clinic will be really valuable to figuring out how we're going to turn this into an actual clinical therapy for human patients.
Do you collaborate with other labs or researchers, scientists on the stem cell?
Oh yeah, absolutely. So here at Hopkins, I've got a handful of very close collaborators. Don Zapp is a world-renowned molecular biologist and also a trained glaucoma specialist, although he hasn't seen patients for a few years. He's so successful in the lab that he's dedicated himself full-time to doing that. And he has actually developed some of the stem cell lines that we're working on. And Johns Hopkins is a great place to be for this kind of work because I've got collaborators in the neuroscience department and the Department of Biomedical Engineering that are all focused on this sort of work. The other thing I'll say is that this optic nerve regeneration retinal ganglion cell replacement is a tremendous challenge, and it's going to be extremely complex. Certainly no single lab is going to unlock the key that makes this happen, and probably not even a small collaborative group of labs is going to be able to do this. So I and a handful of other co-investigators from throughout the country are currently working on putting together an international consortium of laboratories that are interested in this kind of work. It's called the Retinal Ganglion Cell Replacement Stem Cell Therapy and Optic Nerve Regeneration Consortium, which nicely abbreviates to the RESTORE Consortium.
Okay.
So we've already got 70 different laboratories from across the world that are interested in coming together and sharing ideas and protocols and collaborating on individual aspects of the optic nerve regeneration problem with an ultimate goal of eventually putting together all the pieces so that we can come up with a protocol that could actually be implemented clinically.
That's fantastic. What kind of platform do you use for collaboration? That must get pretty complicated. You've got a complicated disease process and then a lot of different people to coordinate with.
Yeah, exactly. So for individual existing collaborations, a lot of it is face-to-face meetings or Zoom conference calls and things like that. For this larger consortium that we're trying to build, we've actually got a three-stage model where we're going to be working together in a consensus-based platform to ask first what are the most important questions and challenges that we need to answer and overcome to make optic nerve regeneration happen. That will be done mostly virtually through web conferences and surveys. We're going to have an in-person meeting next year, actually, right before the ARVO meeting in Denver, Colorado in April. That meeting has researchers, more than 10,000 people typically come from around the world, so we're going to leverage the existing infrastructure of that and have a face-to-face roundtable discussion there. And then after that, we're going to build an online collaborative platform, sort of like a Reddit, that will have discussion boards and collaborative tools for large email threads and things like that so that people can share ideas. And then hopefully there will be individual labs working with each other, conducting specific experiments that come out of this.
So now I have to ask the loaded question every glaucoma patient wants to know. How long do you think it will be before the stem cell, program is able to restore optic nerve function?
Yeah, it's a good question. I get that a lot, and it's really impossible for me to give a well-informed answer on Certainly one way I could say is longer than we would like, but I believe it's not impossible. It won't take forever.
Right.
The problem is that replacing retinal ganglion cells is incredibly complex. I mentioned we're born with 1 million ganglion cells, but they're not all the same. There's probably 30 or 40 different subtypes of ganglion cells that each do slightly different types of encoding and communicating of visual information to the brain, and they don't go to one place in the brain. They go to dozens of different subcortical areas in the brain that all have to do with different types of visual information processing. So in some ways, we don't really even know all of the questions that we need to answer and all the challenges that need to be overcome to make this happen. And right now, our work is occurring in early preclinical stages, meaning like lab mice and lab rats. And so we need to show that we have safety and efficacy in those early models, and then translate that into safety and efficacy in larger preclinical models before we can even think about doing this in human patients. So I think the minimum is, 8 to 10 years probably, but I do think that we're going to get there. And I am a clinician scientist starting off his career, so I've got decades ahead of me, and I firmly believe that this is a treatment that I'll be offering to my patients within my career.
That's fantastic. And you mentioned 70 some collaborators in this consortium. Are they all optic nerve, or is it like spinal cord and all other types of nerve damage?
Yeah, it's all sorts of different people studying all kinds of different things. So some of them study retina, but they don't study optic nerve. They study photoreceptors.
Okay.
And that's relevant because there's actually been more progress and more immediate clinical relevance to stem cell transplantation in the eye for diseases that affect the photoreceptors, like macular degeneration and some types of inherited retinal dystrophies. Those cell types are much simpler and less complex than retinal ganglion cells, and those will probably be trialed in human beings within a few years. I mean, retinal pigment epithelial cells for AMD is under clinical trial in human beings right now. And so, I think we have a lot to learn from our colleagues that are engaged in that work. We have some people that are interested in optic nerve, not necessarily in neuroregeneration, but in why the optic nerve and the nerve fibers die in glaucoma.
Right.
And that's relevant because if we replace retinal ganglion cells, there's still going to be a disease process there. And we don't want our stem cell-derived nerve fibers to die just like the original nerve fibers did as well. And then we have people that are studying neurodegenerative diseases in the brain, and like you alluded to, in the spinal cord. We have biomedical engineers that are figuring out how to electrospin scaffolds and things that can guide the fibers of these cells from the eye to the brain after they're transplanted. So it's certainly going to be an interdisciplinary and collaborative effort. We're going to need help from all realms of science to figure this out.
Okay, well, thank you so much for your time. We really appreciate it. It's been fascinating learning more about this and also helping our donors understand the importance of our continued support for programs like the MAPS program and other research efforts. And looking forward to getting results in the future that will help everybody.
Yeah, no, thank you. And thank you to everyone that has contributed through your foundation. It's incredibly important to the work that I do and the work that many other people do, but I think more importantly to getting new treatments out to the patients with glaucoma and other diseases that can benefit from it.
Definitely.
Thanks very much.
Thank you.